Hello everyone. Today we will be discussing cells, and we're going to be talking about the structure of a cell and the organelles and how we make proteins. We're probably going to divide this up into two different videos and talk about different topics here, but we'll start with the structure and the organization within our cells. So there are three components to a cell. So first we have the plasma membrane, which basically forms the border of the cell. This is how the cell is going to communicate with its external environment, and we'll break down the structure of it in a second, but there are a lot of proteins on the outside which help transport materials in and out of the cell. And then we have the cytoplasm, which is the inside of the cell, the intracellular fluid, otherwise known as the cytosol, and then all of the proteins and organelles that are found within. Last, we have the nucleus, which houses all of your DNA and all of your genetics. So everything from your eye color to how tall you are, this is found in your genetic code. And, most of the cells within your body will contain all of your DNA within that nucleus. When we look at the plasma membrane, we could see that most of the lipid bilayer is made up of what's called phospholipids, and we did discuss this in the chemistry lecture. They have this polar head with a phosphate group, and that is hydrophilic, meaning it gets along well with the aqueous solution, both on the inside of the cell and on the outside of the cell. But then we have this fatty acid tails on both sides. And so what that is going to do, since it's hydrophobic, it's going to basically block polar molecules from just crossing the border and heading from the external environment to the internal environment, or vice versa. And so polar molecules are going to need proteins in order to transport them from the external to the internal environment. We'll talk about that a little bit more in future chapters. But in addition to phospholipids, which make up most of the lipid bilayer, you also have cholesterol that is in that bilayer, and then glycolipids, which are lipids attached to carbohydrate groups. And so since the lipid bilayer has both those hydrophilic qualities and hydrophobic qualities, we talked about this term in the chemistry section as well, but that makes it amphipathic, meaning that it's both hydrophilic, meaning it could react well with water, and it's got a hydrophobic part as well. So within the plasma membrane, we are going to have a lot of proteins which carry out different functions. Some are integral, meaning that they are actually going to be anchored. They're going to have a non-polar some non-polar amino acids that are going to anchor it within this hydrophobic layer, and then communicate with either the external part or the internal part of the cell. And then transmembrane proteins, like this one here, they're going to not only go through the bilayer, they're going to communicate with the external environment and the internal environment. And so these are really good for signaling when there are external signals outside of the cell. It can start a reaction, and it could start functions inside of the cell that they need to carry out. You have peripheral proteins, which are not anchored to that hydrophobic layer. They're just on the external side or the internal side, attached to those polar heads. You have glycoproteins, which are proteins, again, attached to carbohydrate groups, and then glycolaxis, which is found in your gastrointestinal organs, and it's also found in your vasculature system, but it helps in the immune process, and it has many different functions as well. Next, we have the cytosol, which is where all these chemical reactions that happen within our cells are going to take place. It's most of our cell volume and made up of mostly water. And so again, we call that an aqueous solution. It's got many different ions, proteins, glucoses, amino acids. It's got all these different organelles that occur within the cytosol. There's a lot of different materials inside that cytosol, a lot of different reactions taking place. And so, again, the cytosol, that's just the liquid that this is all taking place in. The organelles within the cytoplasm are going to be tiny structures that carry out specific functions. And so cells, it's hard to think about, but it's like a little city. There's a lot of moving parts, and there's a lot of things going on in order for the cell to carry out its function at rest. And so, these organelles will perform specific functions in cellular growth, maintenance, and reproduction, and we're going to break down each and every single one. Ribosomes are the site of protein synthesis. Again, we have tens of thousands of different proteins, and so the cell constantly needs to make proteins in order to carry out specific actions. And so they are made up of what's called ribosomal RNA, and then each one includes about 80 proteins. But this site here, in that ribosome, this is where we are going to be making proteins, or it's the site of protein synthesis. The endoplasmic reticulum is going to have two separate parts, and it's going to play a big role in the synthesis of proteins and lipids. And so again, those two sections, we have a rough endoplasmic reticulum, which contains those ribosomes. And again, ribosomes, the site of protein synthesis. So a lot of the proteins that are made in the cell are going to be produced right here in the ribosomes of the rough ER. And then we have the smooth ER as well, which does not have ribosomes, but is the site of lipid synthesis. So, certain important signaling molecules and molecules in our cells, such as steroids and production of fatty acids, things like that. All these different lipids, that's going to be made in the smooth ER. The Golgi complex further modifies protein made by the rough ER. A lot of times, it's not just the amino acids that can go into a protein. For example, we said that a lot of proteins in the plasma membrane have some sort of carbohydrate on them. So in order to get glycosylated, the Golgi complex is going to help further modify those proteins that were produced by the rough ER. A lot of proteins do need to be modified once they are synthesized. So again, that takes place in this Golgi complex. And once these proteins are synthesized by the ribosomes of the rough ER, what's going to happen is that they are going to be transported via transport vesicles. And again, future chapters, we're going to talk a little bit more about transporting materials inside and outside of the cells. But the membrane of the rough ER is going to be pinched and butted to basically enclose this protein. And then that transport vesicle brings it to the Golgi complex. And then once those proteins enter the lumen of the Golgi, they are modified, a lot of times glycosylated, depending on what the function of the protein is. And so once they get through the cisternae and then need to be transported again, a similar process is going to occur. There's going to be a budding of this Golgi membrane, and then it's got a few options that newly modified protein can take. Sometimes the protein will be transported to the membrane, so a lot of those glycosylated proteins we talked about, so they are going to be transported to the membrane and then anchored there for their function. We have some secretory vessels that will take that protein, and if it's meant for a function outside of the cell, it will be transported through exocytosis, and that's basically the transporting of a material from the inside to the outside of the cell. Or a lot of times those proteins are just broken down further by lysosomes, and then those amino acids and those parts of that protein can be further used in other processes. Ever since grade school, you've heard the expression, the mitochondria is the powerhouse of the cell. And, you may have the question, why? And the reason why is because it makes basically a lot of the energy found within our cells. And so that's why it's the powerhouse. It's supplying that molecule we talked about last time, ATP. And again, that is the energy currency. If you want reactions to happen, if you want functions to take place inside the cell, a lot of them are going to require energy. And so the mitochondria is going to be responsible for making that ATP. And that's why we call it the powerhouse. And so in future chapters, we'll talk a little bit more about aerobic respiration and how that ATP is made. But again, that mitochondria, that's going to be the source for a lot of the energy within the cell. Again, a cell is very similar to a city, and you need like a bunch of moving parts and a bunch of different workers to carry out certain jobs. So lysosomes, peroxisomes, and proteasomes, you can consider them like the cleanup crew of the cell, right? So lysosomes are going to be these vesicles, these membrane enclosed vesicles that contain digestive enzymes that digest a wide variety of substances. And so whether it be, certain types of microbes or, proteins that we don't need anymore, or, just a bunch of different materials that the cell needs to basically break down and then recycle. So those lysosomes are going to be responsible for that. We also have peroxisomes, which are a little bit smaller, but again, contain certain proteins, in this case oxidases, that are going to help metabolize amino acids and fatty acids. So then we could break down, those materials and then use them in the cell. And then we have proteasomes, which degrade unwanted or damaged proteins that are found within the cytosol. And again, the cells are constantly building proteins, constantly carrying out reactions. So these things are all important. This way, if we need additional amino acids, or we need to get rid of the proteins that are no longer working, these organelles are going to be very critical for those functions. So the cell has a network of what's called the cytoskeleton, which are protein filaments that are basically going to help in structure and movement and function. So there are three main types. We have what's called microfilaments, intermediate filaments, and then microtubules. Microfilaments, otherwise known as actin filaments, are the thinnest out of all the cytoskeleton proteins there, and they are most prevalent at the edge of the cell, and they help form out and develop what's called the microvilli, which are these like hair-like structures that a lot of times will help communicate with the external environment. And this microfilaments will be important to help the cell move, and then also to provide some mechanical support. Intermediate filaments are a little bit bigger, but they're going to be found in places where the cell encounters stress. And so, it's going to be very important around the nuclei, and it's going to help those organelles all stay in a proper position. And it's also going to be responsible for tethering cells from one to the other. Microtubules are going to be the largest out of the cytoskeletal components. They are these long hollow tubes made up of gamma, alpha, and beta tubulin, and they are going to be important for forming the shape of the cell and the movement of organelles and certain materials. And so what you're going to have is a lot of transport proteins, which are actually going to walk along, well, I say walk, but they're going to move along this microtubule, transporting materials from one side of the cell to another. The centrosome is going to be where these microtubules are going to originate from, and I believe that's probably the most important thing here on this slide. We will talk about cellular division in the next video, and we're going to see it's from these centrosomes that centrioles are going to come out and be able to divide the cell into two different ones. We have little hair-like projections, which are called cilia and flagella, and they are very important, again, for the communication for the cell to the external environment, and also, in the case of the flagella, are very important for movement of the whole cell. Last, we have the nucleus of the cell, and this component is where all of your genetic information is held. And those genes are basically a coding in order to make proteins. So whenever we need to synthesize a protein, we're going to need to read that genetic material in order to form that protein. And we're going to dive into a little bit how those proteins are expressed. Like the other organelles in the cell, the nucleus is going to have a membrane, and since the nucleus is such an important site because it holds all that genetic information, it needs a good membrane in order to protect it. And so we call that the nuclear envelope here, which is the double membrane that surrounds the nucleus. So again, you just don't want a lot of materials just coming and going as they please in and out of either the nucleus or the cell. And so, you need to form these barriers in order to protect the cell and the nucleus. And so in order for certain materials to get in and out of the cell, however, there is nuclear pores, which are going to help transport different materials in and out of the nucleus. The nucleolus is an area of the nucleus where there is a cluster of DNA, RNA, and other proteins, but it's very important for the synthesis of ribosomal RNA and for the synthesis of ribosomes, which are going to be used for protein synthesis. DNA is a long strand of all this genetic information, of all these nucleotides in a sequence together. And so if you took one cell's DNA, all the DNA within one cell, and just extended it out to one long line, it would actually be over 6 feet tall, which is pretty amazing considering that it's just one cell. And so we need to basically compact all of that DNA together in order to store all this genetic information. And so this DNA is wrapped around proteins called histones that you could see right here. And so all that DNA strands will be wrapped around these histone proteins and contracted together to make chromatin, right? And so this is how all your genetic information is stored. And when we need to make a protein, we'll unlock a gene around that histone in order to read it, but a little bit more on that later. Before a dividing cell, what we're going to need, since we are making a new cell, is for a set of that DNA for the original cell and then a set of the DNA for the next cell. And so when we divide, we develop these chromosomes, which are ultra condensed chromatin, and then we're going to have two sister chromatins which are going to be split apart so each cell can have its own pair of DNA. Again, the cell has to make many proteins in order to function, and so that process is through what's called genetic expression or gene expression, and that definition is when a gene's DNA is used as a template for protein synthesis. And it involves two very important steps. Transcription, where the DNA is used as a template to make something called messenger RNA, and then that messenger RNA is going to code for how to make the protein. And so the ribosome is going to read this messenger RNA, and from there we're going to get translation, where messenger RNA is used in order to make proteins. This is the video that is found in the slides. I typically don't do this, but I'm just going to speak over it a little bit to further explain what is going on during genetic expression. Again, all of this genetic material is going to be stored within the nucleus, and that nucleus contains genes, which are basically codings for different proteins. And so, that DNA is going to be the template in order to make the protein. And so in order to do that, all these genes are typically, again, wrapped around a histone. All this, genetic material is all wrapped around. And so we need to actually unlock it from that histone so then we could read those genes and then express those genes. So transcription, again, is using DNA in order to make something called messenger RNA. So we're basically transcribing the code. And so a protein here that's popping up called RNA polymerase is going to bind to a promoter region, and then RNA polymerase, as it reads that gene, is going to attach nucleotides that match the DNA, right? But this is messenger RNA. So the only difference is that when it reads over a adenine, it's going to place a uracil there instead. And so now we see this messenger RNA, and that's going to be used in order to basically be a code in order to make a protein. Once we have that messenger RNA, it needs to be transported to the ribosome. So we talked about RNA in the chemistry chapter, but really didn't break down the different types of RNA. You see messenger RNA again, which is going to be used as that information to synthesize proteins. Transfer RNA is going to be used during the protein synthesis process in order to transport amino acids and link them together. And then the ribosomal RNA, that's going to be used to make up the actual ribosome. What's going to happen is that this messenger RNA, once it's transported to the ribosome, it's going to basically feed that information through the ribosome. And so the messenger RNA is going to have groups of 3, and these are called codons. So what's going to happen is, as this messenger RNA is being read, a transfer RNA with matching nucleotides is going to bind to that codon. And then that transfer RNA with that specific codon, with that specific code of 3, is going to have a amino acid. And so what's going to happen is that once then another transfer RNA comes and binds to the next codon, There's going to be a linkage formed between these two amino acids up top. And then now this old transfer RNA will be booted out of the ribosome. And speed this up a little bit. And now the messenger RNA comes through. Another matching codon will be put right next to it. And then that amino acid joins the chain. And so here's how you get the actual translation. So we're going from this code, this mRNA, to actually now linking together amino acids to form a polypeptide chain. And so then once that polypeptide chain is formed and it's going to keep on building, it's going to be long enough and it's going to form, this secondary structure, which we talked about, either an alpha helix or a beta sheet. And then all of those come together to form that 3D tertiary structure, right? And so then eventually we're going to get this 3D shape. We're going to get something that resembles the protein's final form. And then a lot of times, like we said, it needs to be modulated. So like it showed in this example here, it's glycosylated. So there's chains of glucoses being added to the protein, and that's going to help it. A lot of times, if it's a transmembrane protein, be anchored. Or it's going to signal where that protein should go. So again, I don't want to go too far into the processes that actually occur within the ER and the Golgi apparatus. But for genetic expression, in order to make those proteins, remember the two important things, those two important steps: transcription, turning DNA into mRNA, and then translation, which is actually translating that genetic material, that information, that mRNA, into an actual protein. This is where we will conclude the first part of our lecture. In the next lecture, we will be discussing a little bit more about how cells divide, and then we're going to be talking about the different tissues which are found within our body and the junctions that hold cells together. Thank you guys so much, and please look out for part 2 of this cell lecture.